All right, pleasure to introduce Kathryn Hess from the FL in Switzerland. We'll be talking to us on topological analysis of digitally reconstructed microcircuits.
Thank you very much for the chance to give this talk in this unusual setting. I've never given a computer-based talk before, so we'll see how it goes. And it's a pleasure to talk to you all around the world, I suppose, about this. So the project I'm going to be talking about is work that I've been in, well, in a deep last year or so with people from the Bluebrain at EPFL and together with, let's see if I can, how do I switch my, okay, so together with Pavel Dlotko, who's now at INRIA, and with Ron Levy at the University of Aberdeen, two postdocs, Martina Scolomero and Kate Turner, and with Henry Markham, Edith Mueller, Milte and Michael Reimann from the Bluebrain project. So it's been really great to have such a diverse group of collaborators. It's been really important as well. The only way to actually managing in the neuroscience elements of serious statistics, topology, topological analysis. It's really been a big collaboration on this project. Explaining something about what the Brain project is and what's involved in this particular reconstruction, and then talk about the kind of analysis that we've done of what the simulations have been produced by the Bluebrain project. So this project is doing is making some sort of computer reconstruction of a very small part of the cortex of rat to try to figure out all the very complicated connections that there will be in this microcircuit. What they're doing is about 2-week rats, sacrificed them, as they say in biology, and then took samples from their somatosensory cortexes and looked at the different layers in the cortex. There are 6 layers, labeled from 1 to 6 from the outside to the inside. And they looked at the heights of the different layers, the proportions of the different cell types, and the densities of these cell types in the different layers for 5 different rats, and uses this input data to a certain algorithm that uses biologically motivated organizational principles to reconstruct the circuit. More details on the next slide about exactly how this process goes. So in the end, they made 42 of these microcircuits, which make these sort of hexagonal columns that are layers high. Each microcircuit has about 31,000 neurons in it, and they form roughly 8 million connections. Now, a connection is not just given by one synapse, one touch between an axon and dendrite, but you need actually several synapses that join 2 different neurons to make a connection between 2 neurons. So they made 42 of these, based on each individual rat and 7 on the average data for the rats. They could perform simulated activity, either spontaneous or stimulated activity, on these microcircuits, and with that they could reproduce certain in vitro and in vivo experiments quite well, so they have validation without doing any particular parameter tuning. So it's a wonderful toy online. If you look at the web address that's on this slide, there's what's called the Neocortical microcircuit portal, which is a really great interface to figure out exactly how, what these different layers look like, how many neurons there are in each layer. The number of the neurons in each layer varies a lot. There are 300 in the first layer to up to 12,000 in the 6th layer. Most have several thousand different neurons, the different ways they connect, different neuron types, and so on. 55 different morphological types of neurons, and if you take into account the electrical activity of the neurons as well, there are 207 types. So these columns themselves are about just 0.3 cubic millimeters, really not very big. The height is about 2 millimeters, the radius is about 0.2 millimeters, and these are just little pieces of the cortex, but the structure is already incredibly complex. So I'm going to say something more about the workflow and the production of this microcircuit. So we look at panel A. Sorry?
Can I quickly interrupt you? Can you cue how these, so the 42 microcircuits, how are they arranged? Maybe it's going to be on the picture here.
Each is an individual, so they're not arranged, well, so with 7 that they made for each rat, they made them, since they're hexagonal, they can make them with a center column and then 6 on the outside. They can make these sort of mesocircuits, as they call them, that are built out of 7 microcircuits. Each microcircuit is really sort of an entity unto itself.
6 layers deep?
So 6 layers. If you look at the slide that's currently visible, so you can see, maybe if I explain what's on this slide, it'll be more clear.
Okay. Thank you.
So if you look in the first panel, panel A, what they do is they have these 55 different morphological types of neurons, and they have certain specific examples of each morphological type that are taken from actual cortical slices that have been traced, and they data about what these neurons actually look like. So they have examples of them, and then they can clone them, making small tweaks here, so they have some variation. They have these samples of each morphological type, each of the 55. Then they go into the columns, and you have the 6 different layers in columns, 1, 6 like this, and they know which sort of neurons are supposed to appear in which layer, and what are their densities, and where their placements are supposed, roughly supposed to be. And so they neurons like that, and then they reconstruct the connectivity. So it's a complicated process that involves first just saying, okay, it has to be close enough in order to form a synapse. And then they look at that, they create way too many synapses, and there's a complicated 4-stage pruning process that they go through in order to reduce the number of synapses to something biologically readable. And then they look at the different level types of the neurons, the different types of the synapses, and so it's a very complex reconstruction they do. And in order to any kind of experiments for activity, you have some sort of way of getting an input stimulus. So they have something that simulates thalamocortical input with different looks like well. And then they could test either spontaneous or stimulated activity. So I'll discuss more about activity in just a moment. This picture here is just a visualization of the microcircuit where we see only the somas, so the cell bodies of the neurons and their dendrites. And then it's color-coded as well by morphological type, because there's certain morphological types that are some layers than others. One nice picture that's come out of the instructions. Now, about activity, because you don't want the network just to sit there. You're not interested just in sort of the connectome itself and how the neurons are connected, but you want them to do something because the brain is active, right?
Kathryn?
Yeah?
Can I interrupt you again? So what part of this is experimental and what part of it is a model? So it's
a lot of experimental data that's into the reconstructions. They have they're very deep search of the literature, deep and broad search of the literature, to find out everything they can about all that is known from wet lab experiments about how different neuron types connect, what their synapses look like, how their synapses behave. So there's a lot of input data from doing different kinds of experiments like this. And but there are way too many possible connections. If you think about 55 squared possible connections you have to understand, and not all of those, a very small portion of those has actually been studied deeply in the lab. So something they had to extrapolate based on certain biological physical principles, and other things have been biological input. Some of it is just based on physical principles, and so it varies a lot. And then they go through and there's some experiments data that isn't input into the reconstruction, and then they use that for validation. They run experiments on the digital reconstruction to test against biological data.
At the macro level, I guess.
Well, just at the level of this size, because you don't have.
Yeah, that's what I meant by macro. Yeah.
Okay.
Thank you.
You're welcome. So, as I said, in order you want the point of having a neural circuit is for it to be active, and even when you're not conscious of something, there's always some sort of activity bubbling around in your brain, and due to polarization of the neurons and so on, And depending on the calcium concentration in cerebral fluid, you have different levels of activity. And so they have stimulating different concentrations of calcium, and the spontaneous activity that's elicited from the digital reconstruction depends on this calcium concentration. So here we see another example of one of these optimized circuits with its different layers that are labeled. And the color coding of the neurons is telling you something about their level of activity, whether they're hyperpolarized, depolarized, or in the process of spiking, actually. Okay, so the spontaneous activity. And also want to be able to stimulate that activity. So the circuit is made up of a lot of so each circuit is made up of 310 mini columns, which are made each of 1,000 neurons. And so they know that if you look at the actual brain, then you're roughly going to have one fiber in from the thalamus in each microcolumn like this. So they have one stimulated incoming fiber for each one of these mini columns, and then they can stimulate activity by stimulating pulse of electricity that goes through this fiber and see how it interacts with the synapses and so on. And to simulate something like flicking the whisker of a rat, because if you flick the whisker of a rat, it's going to create some sort of reaction in the thalamus, which is then transported to the somatosensory cortex by these fibers. And so it's a kind of activity that you can simulate with these microcolumns. Okay, run experiments if you want to on this. So the sort of validation that you can do. So in panel A, it's just one of these virtual brain slices built up from looking at a bunch of columns next to each other. And if you want panel B, then showing what happens in spontaneous activity when you have a high calcium concentration or a simulated high calcium concentration, and you see that all of this is a very high degree of coordination and the spiking among the different neurons. And then you look at panel C, then you're in low calcium concentration, and you see it's kind of random, kind of just activity. And D, what they have is a multi-electrode array that's applied in vitro to a 300 micrometer thick cortical slice. And they're actual biology, an actual wet lab and in vitro experiment, and they're trying to see what happens with the spiking with high calcium and low calcium, and we see a very similar sort of behavior that you have in an actual in vitro experiment compared to what is happening in silico. So sort of validation has been done on this model. Okay. Why something like this? I mean, sort of this, the beauty of the gesture, as you'd say in French, but also, I mean, there are interesting applications. So really interested in this sort of emergent structure and property of the circuit, coming from knowing some basic rules about how things are supposed to be put together. You want to see what sort of structure emerges for the whole. You know, it's the sort of thing a topologist love, going from local to global. We know locally how the different neurons interact, and we're interested globally in what the structure of the circuit is supposed to be. So the sort of thing you can deduce from this are what sort of connections and synapses you get between different neuron types, because all of this is known. The number and combination of pre-synaptic, so the presynaptic is the neuron that's sending a signal, postsynaptic is the one that receives it for the different neuron types. And then the layers, the neurons, the connection types, all of this interacts with the dynamics of the network. And then they can play with the extracellular calcium concentration, which, as we saw, affects how what the spontaneous activity looks like, either highly coordinated or chaotic or something in between. So what are my collaborators and I interested in when we're looking at this? Well, what's right? So, I want to see what is anything special about the topology of this microcircuit, and what is experience for the neuroscientifically be. So, I mean, topology of the microcircuit, what do I mean by that? I mean, for the time being, we've been thinking of it really as a directed graph, where the vertices are the neurons themselves, and you have a directed edge from one neuron to another, if there's a connection between the two neurons, so if a direct neuron A to neuron B, if neuron A is the synaptic neuron and you have a signal that goes to a postsynaptic neuron, neuron B. So this thing is a directed graph, and we want to, in various ways, associate to this directed graph and see whether there's anything interesting going on. Well, graph theory has already been applied a lot to seeing the networks in general, and topology is sort of the next step beyond graph theory, and as most of the audience here probably knows, it's of course for notions of proximity and particularly connectivity, which we're very interested in for this network. Local, a very important thing that topology can do, and that's what this microcircuit is about, taking local properties of the neurons and looking at the emergent global properties of the whole circuit. We're interested in sort of higher order connectivity, not just is neuron A connected to neuron B, but in how many possible ways and going through how many possible other neurons. And that's a great, topology is really good at telling us things like that too. I'm going to go briefly through the topological toolbox that we use in order to study this network. And most of this will be known to most of the audience, I imagine. With the fact that we really take direction into account in a very serious way, because it's really important that we think of this directing the microcircuit as being directed. It's really important to know in which direction the signal is flowing through the network. So what we use instead of just usual simple complexes, we're going to think of ordered simplicial complexes. With an ordered simplicial complex is just a collection of finite ordered sets, or lists, if you will, such that if a ordered set belongs to my simple complex, then every subset as well, and it inherits an order from the order sigma. Look at the example here. This example, I have all the sets, subsets of this list 1, 2, 3, and so that's going to be an abstract ordered simplicial complex. If I look at the second example, then all of a sudden I have this 3, 1. Which said if I look at this list with 3, it's now missing the sublist 1, 3, so this is not going to be an ordered simplicial complex. On the other hand, if we let this one go, we drop the top simplex, or the top set, then it is an ordered simplicial complex, no problem. The examples, look at this more metrically in just a moment. So we're really going to take action very seriously and think about these ordered simplicial complexes.
Okay. This is then I take it's something more structured than you get from just inducing an orientation on a simplicial complex for ordering the vertices. I think if you have a simplicial complex and you order the vertices, then yes, you can get something. We really want to keep this order in mind, and we'll see in a moment that if you think about that more geometrically, you have to be very careful about what this order means. In the next, I think it'll be a better answer to your question. Okay. Okay.
We're going to associate any one of these abstract ordered simplicial complexes a topological space, which would be, we'll think of metric realization, by associating to every abstract n simplex, so every ordered list or ordered set with an element, we'll associate a geometric n simplex, and we're going to glue them together along their faces, respecting the orientation, respecting the order. So in the end, you can, the logical space, you forget the order and you just have a, but the order is, or the orientations are telling you how you should glue things together. The reason, so this is going to be the next one. The reason we're interested in thinking about these ordered simplicial complexes is because we want to work with directed flag complexes and not just ordinary flag complexes. So if you have a graph, it's a trick to consider the simplicial complex that's called the flag complex or the click complex, where n simplex is an n+1 click, so an all-to-all connected +1 vertices in your graph. But here we're thinking about directed graphs, and we want to take the notion of direction seriously when we think about clicks. So we want to think about sort of directed clicks. So let's start with a directed graph. So with the vertices V, a set of edges E, and then tau, which is going to edge to the pair, which is its beginning and its end. So we now have a sense of direction. Associate to any directed graph like that is directed flag complex. So the set of zero simplices is just a set of vertices. The set of n simplices will be the sets of n plus 1 vertices, set an ordered set of n plus 1 vertices. So that look at any vi vj, where I is strictly less than j, then that is an edge in my graph. To account the sense of order these ways. N simplices correspond to directed n plus 1 clicks.
Question. So is there an example of a collection of edges that would translate to a click directly that do not form a simplex?
Wait, slide is coming. It's definitely coming. Okay. So I want to add one more before we go to this next slide. So the important thing is that every simplex is therefore characterized by an ordered sequence of vertices, but not a set of underlying vertices. So you could, if you have the list V0, V1, V2, and V0, V1, V0, V2, they can both be two simplices and they're going to be distinct. So a way of, okay. And that gives us a way of limiting. So, example of a directed flag complex. So on the left, there's an oriented graph, and then we have its directed flag complex on the right-hand side. If you look in particular at so we get the directions. This is indeed a 3-click, but it's not a directed 3-click because it's exactly this example that I gave before, with we had 1, 2, 3. That here I have 1, 2, 2, 3, but I have 3, 1, and not 1, 3. Number these 1, 2, and 3. And this doesn't get filled in. There isn't actually a 2 simplex here. There isn't an ordered 2 simplex. There's 3 ordered 1s that are 0. Later, okay. And on the other hand, if we look at this one here, 1, let's see this one, 2, 3, this here, this is a direct click given by guys, and so it does get filled in. And in fact, whole here is a directed 4-click, and so it's filled in with the 3 simplex. This is more it was more useful for topologists to think about that. Since there was an edge missing, of course, there wasn't too simple. So what we do when we take this directed graph is then we associate to it its directed flag, and then we can take its geometric position and get a topological space, and we associate different invariants to this simplicial complex. And I'm just going to let Ck of S be the number of K simplices. And just say that, hey, we have an n-dimensional complex, so there are no simplices for K greater than n. So the Euler characteristic in that case is, of course, the alternating sum of the numbers of simplices. And this is important invariant to compute. I always kind of underestimated the Euler characteristic until I heard a talk by Robert Adler last year in which he said, Euler characteristic contains a lot of information, and he was exactly right. So really interesting invariant to study. So when we're looking at the Euler characteristic of the directed flag complex, then we're also interested in the Betty numbers, which we're going to take the mod 2 Betty numbers because that's what computers can deal with the most easily. And so there's that should be a K. Amazing, get rid of all of the typos. So look at the Kth Betty number, which is going to be the dimension of HK with the F2 vector space. As everybody on this talk knows, the Euler characteristic is actually also equal to the alternating sum of the Betty numbers. And so we're going to we'll start with this directed graph that we're going to get from the microcircuit, look at the associated directed flag complex, and we're going to look at its Euler characteristic and its Betty numbers and see whether they say anything interesting at all. So here's a little sample computation with some sort of directed model of the two-sphere. Okay. So now let's talk to the microcircuits. So is microcircuits. We're going to look at the underlying directed graphs and analyze them. You can say about the topology. So of this reconstruction is, of course, more than just a directed graph. There are all sorts of extra weighting. There's all kinds of extra information that we're completely ignoring for the time being, and we hope at some point to take this into account when we do further work that we do. For the time being, we're just going to look at this thing as a directed graph, look at the binary adjacency matrix that gives this graph, and generate the associated flag complexes, and do this for all of 42 the reconstructed microcircuits, and to have some kind of comparison with other sorts of graphs. I mean, the comparison we did was with Ardèche-Régnier random graphs, a random graph with 31,000 vertices, and said, well, we'll take the connection probability in this Ardèche-Régnier graph to be that of the average connection probability in the microcircuit. And of course, didn't expect it to be the same. We wanted to see how different it was from the microcircuit. It's not a very interesting comparison to make. You expect these microcircuits to be far from them to that extent. So we did a few more sophisticated comparisons. In particular, we took the average microcircuits and randomized it partially so that you could either distance-dependent connection probability either between different layers in the microcircuit or between different morphological neuron types, randomized. So it was not very far from being the actual microcircuit. The neurons were all placed as before. It's just that a little bit how these were connected by randomization. The test we did was comparing these microcircuits to circuits that were obtained by something called Peter's rule, which is kind of used as a model for how neuromicrocircuits are formed. I mean, neurons will say, oh, no, we know this is ridiculous. This isn't actually a good representation of reality. But nonetheless, this is, sort of a first approximation. And one reason that we did this comparison was that people have the misconception that these blue microcircuits are actually constructed simply with using this Peter's rule. So we said, okay, let's actually do Peter's rule, which is just saying, okay, we're going to keep the neurons placed as they actually are in the reconstructed microcircuit, but then have a much simpler rule for how to form the connections. If the axons of one get within 3 micrometers of the dendrites of the other, then we will form a connection, and then prune them sort of randomly with the uniformability until we get a number of connections. So much less sophisticated in the sort of pruning that they actually do in constructing the blue brain microcircuit. So we did 42 actual microcircuits, and then we did randomizations and did various comparisons. So what we did was to first look at these directed flight complexes and just look how many simplices they have. So what we were counting were numbers of these directed cliques in the graph. A reasonable thing to compute. Why should this be biologically interesting to think about it? Well, consider what you have. We have one of these directed cliques. Standing at a set of vertices, a directed graph, which has a source and a sink. Have all the information flowing from the source to the sink. And having a large clique means that you have a very robust network, basically, of information flow from the source to this sink. And somehow, the neuroscientific idea is that these sort of directed cliques should be somehow functional units in a microcircuit, that they should work together. And in any case, if you're trying to cause a neuron to fire, to spike, then you more than just one spike come another neuron. You need a lot of incoming spikes, and so it makes sense that you're interested in having these sorts of large cliques that we're converging a lot of information onto one point. So this is why neuroscientifically it makes sense to be interested in looking for these large directed cliques. So what we see when we actually did the computation, let's see, let me say some more here. A few words about some of the work that Euler really did for us. So getting how to generate and store these directed flight complexes. We have Hasse diagrams as a form in which to store these. And then we're counting the number of simplices in each dimension, so the number of correct directed clicks of the different sizes, and computed the Euler characteristics. Look to see, okay, what's the homological dimension of these like complexes? Wanted to see what the numbers were. And so the complexes are enormous, as I'll show you in just a moment. So we can't actually compute Topology all the way through, but we can calculate the homology in the top degrees by restricting to co-skeleton. Okay, so about the distribution of the simplices. So this diagram, in this graph here, we have showing along the x-axis the dimension of the simplices, on the y-axis the number of simplices. So it's as if, for example, with the blue curve, there's nothing after dimension 5, but it's just that the number is so much smaller than 10 to the 7th that they look like 0 on the scale. So maybe you should look at it logarithmically instead. But in any case, the point is that if you look at this, then the blue line is one of the microcircuits. So how many simplices it has. The shading is just for the various standard deviation. What we see is that the microcircuit has a lot more directed clicks, a lot of these simplices than any of the graphs, even the ones that are sort of quite logical, like the ones that preserve the distance-dependent connection probabilities between morphological types or between layers, or even this infamous Peters rule that they apply in neuroscience. And so a lot more than in the graphs. So one point I want to make is that, so my scale, of course, has results about, you know, if you have certain connection probability and you're looking at a random graph, not directed, but undirected, then know something about what the homology is supposed to look like. In this case, a directed graph would be really interesting if somebody would, you know, prove like maps for directed graphs as well. Yes?
What dimension here on the x-axis?
So the dimension of the simplex. So simplices, one simplices, two simplices. So for example, what this says is that you have roughly 80 million two simplices in the average, in the directed flight complex in the average microcircuit.
This is for a fixed simulation of microcircuits, certain rules with a fixed number of vertices.
Right.
All the edges are according to these models.
That's right. So it's the microcircuits that are produced by Blue Brain. So the blue line, appropriately, is the reconstruction that's coming from Blue Brain. And other lines are these other randomizations that I mentioned a couple slides back. So the comparison with those. So for example, with the green line, we have the Ardèche-Régnier, and we see that it has roughly million two simplices. If you take an Ardèche-Régnier graph with 31,000 vertices, connection probability 0.8%, and then, but it has again practically zero, three simplices, whereas the average microcircuit has roughly 70 million number of vertices at the Ardèche-Régnier.
And they've been normalized to have the same number of edges, it looks like?
No, not the same number of edges, just the same number of vertices. The existence of an edge, for example, in the Ardèche-Régnier is, you know, if you have two vertices, the probability of existing an edge is 0.8%.
I see. Just on your graph, they both have the same coordinate for one dimension.
Maybe they do.
So
that makes sense because the probability was chosen so that it was the average connection probability across the microcircuit. So it's not surprising that they have the same number of edges. Yes, it normalized so they have the same number of edges because the average connection probability of the microcircuit is the same as the chosen connection probability for the Ardèche-Régnier graph.
Okay, you just hid the number of zero simplices, that's why it's 0.
The number of zero simplices is 30,000 in both cases. So 31,000 is actually very small compared to 10 million, so it looks like 0.
Okay, all right. Okay. Thank you.
It's because it's a scale problem. Okay.
So, do you have any thoughts or ideas on why the tool and the two distance-preserving simulations are so similar?
I think probably because they're both just distance-based connection probabilities, right? I mean, Peter's rule is a little bit different because it starts off being a distance-based connection probability. If they're close enough, well, not exactly. If they're close enough, they connect, and then you do this thing, which is just a random pruning with a uniform probability. So maybe it's not too surprising that they end up similar in any case. So, this. So we don't see it in this graph because the numbers are so much smaller than 10 million. But we actually have directed clicks of up to 8 neurons in the microcircuits. And there can be of 100 of these things. And both the size of these directed cliques and their numbers are much larger than what was expected. And they emerged simply from this reconstruction rule applied for building the microcircuit. And of these, as you see from the graph, the distribution of the click size is different from those random graphs, even those that are sort of biologically based. So when I was presenting this, people said, well, what sort of neurons are showing up? Where are these large clicks and so on? We looked at this more closely. This is the sort of information we can extract easily from the reconstruction. Have some interesting structural results. So these 3 pie charts show for the 5 simplicities, 6 simplicities, 7 simplicities in the direct flag complex, which layers are represented. So you see that the 5 simplicities, for example, are built primarily of layer 6, but there's also some layer 5s and layer 4 and some layer 3. When we go all the way to the 7 simplicities, so these is 8 clicks, then there's really only layer 6 and layer 5 that are involved. That tells us that those neurons are really the ones that are connecting to lots of others and forming these sort of sync depths. Divided into 2 types. There are excitatory neurons and inhibitory neurons. And I believe the inhibitory neurons are roughly 10% of the neurons in the brain. But in the very large simplicities, the inhibitory neurons really predominate. In the 7 simplicities, they are entirely made of excitatory neurons. So very few inhibitory neurons that are represented in these high-dimensional simplicities. It kind of makes sense because the inhibitory neurons are there to sort of stop activity, stop things from moving. Somehow sense. And which morphological types show up in these large directed clicks, then there are certain morphological types that are really represented compared to their density in the population of all neurons. So there's one that are what they call the untufted parental, the UTPCs in layer 6 that make up 29% of all the neurons in 6 simplicities, but that are really only 15% of the layer 6 excitatory neurons. So this is, for me, this is botany for the time being. This is just, examining and saying what is there, and then we'll try to figure out whether it really means anything. But at least we see that it's sort of the patterns that are coming up, the structure effects. Okay, say a little bit about Euler characteristic. So the point of this slide is really to say that when we use reconstructions, for each individual rest, they take into account this biological information. As I said, the heights of the different layers, the proportions of cell types, the cell densities, and so on. And we see that this biological input data actually does have an effect on the structure, because you can see that the characteristic, actually here we have multiples 10 to the 7th, does vary a lot. We seem to have sort of 2 different types here. Basic characteristic. Rats 2 and 3 are similar, and rats 1, 4, and 5 have similar Euler characteristics. But there's a very difference between these different types. So what I'm representing here are the 7 different reconstructions for each rat. For homological dimension, so it turns out that there's homology up through dimension 5 in each case. Some of the numbers are kind of small, so here we're really on a scale from 10 to 80. There are no longer powers of 10 that are involved. And there can be some variability with a particular rat type. But we at least see that there are really quite high-dimensional homology classes as well, which if you compare this to, for example, Matt Kale's results about where you can find homology in Renyi graphs of this size, I mean, you wouldn't expect to find anything. dimension 5. So quite complex structures in this graph if it's able to produce functional homology classes. Yes?
What?
So these 5 different rats as to produce input data.
So from experiments.
Actual experimental data, yes.
And
actual rats.
So they use those to construct the model or use the parameters for the models?
Basically, input parameters which were given by these layer heights and cell densities and so on that they input into algorithm and they run it and make their reconstructions. So they did 7 reconstructions for each rat. That's why there are 7 different points there represented for each rat.
And
Kathryn?
where the different reconstructions vary?
So they just take the input data to any of the reconstructions. They need basic information about what sort of morphological types show up in the different layers and the relative densities of the different morphological types and how what's actual height of the different layers is. And so they take that for the 5 different rats, they gathered that data once they were sacrificed, and then they could input that into this reconstruction. And there's a certain quite degree of stochasticity in the reconstruction. So even if you put exactly the same parameters, you're going to get a number of different variants of the reconstruction. So they have 7 different variants for each rat.
I see. So it wasn't that there were 7 different kind of slices taken out of the brain.
That's just due to the stochastic element of the reconstruction.
I see. Okay.
You're welcome.
Kathryn?
Yes?
On the slide, you had these 1, 4, and 5 being very similar, and 2 and 3 being kind of similar. Groupings correspond to anything biologically? Are there rats from known strands of rat or something?
This is, the sort of biology where they have these very purebred rats. So there's a very specific type of rat. They're basically clones, as far as I can tell. And so it's just coming from the fact that they have this different input data that's causing. So we don't know. We keep wondering whether we could find the lab that actually sacrificed the rats. That was one of them particularly intelligent, or was one of them, but because that's a difference in the topology of the networks. But what it means yet. But to conclude this discussion of homological dimension, I'd like to point out that if you look at the flag complexes that are coming from Renyi or from Peter's rule, then the homological dimension is in fact 2 and 5, so it's a lot less complex structure. For the other types of random graphs that were a little more biological, perhaps, they are at most homological dimension 4, but if they do have some homology dimension 4, it's very, very little. Just like the dimension is maybe 2 or 3 or something like that. So those are also much complex in structure. So a little bit, in the next episode, talking about how we could analyze functions. So this is just looking at structure. And, we know yet what this structure means, but at least we see that there is some sort of complex structure there. And our hypothesis is that it actually has some functional role as well. So see whether topology could detect anything about actual function of the network and not just its structure. Let's look at the problem where we had different input, so coming from these neocortical fibers through the same column that had very similar firing rates. So, how often you had neurons that were actually firing, but where the stimuli were really different. And to say, well, okay, can topology see the difference between these things, even if something sort of the ground firing rates can't? What they did to create 2 kinds of stimulus. One causing synchronous spikes near the center of the microcircuit, another which was causing synchronous spikes on the periphery of the microcircuit to make shapes that were of a form of a point or a circle. And I'll show an illustration on the next slide. So this is similar to something they did in this paper by Mark Romani, which I mentioned on a few slides here, which you can find in Cell. It's a really, really fantastic paper to read. So they sort of stimulated whisker deflection as I talked about, and they're just trying to ensure that we have the same average firing rate, at least to be. So repeat the stimulus every 50 milliseconds for a total of 1 second. Did 20 trials of each stimulus and did various comparisons. There is trial-to-trial variability in the result in the reaction to the stimulus due to the stochasticity that's inherent in the model. Here's describing the experiment. So if you look at panel A, what we're looking at is a heat map of the firing of the neurons. So we're sort of looking at the column from above, and it's clearest, I think, in the boxes that are labeled 5 to 10 milliseconds. You see the form of a circle and the form of a point, which is where the stimulus is incoming from the fibers. And the heat map shows how this diffuses through the column. 35 milliseconds, and then you're going to input another stimulus 50 seconds. Those B and C are just showing you what's going on with the firing. These are what are called raster plots, where you're looking at where the neurons are firing. And so we see the neurons firing at time 0, time 50, roughly time 100, time 150, etc. And basically a histogram derived from panel B. And then in panel D, what we see is the mean firing rate in hertz for these two different stimuli, the circle and the point. And the little diamonds are showing us where we put the stimulus. So we see this interesting sort of hysteresis effect that after the first stimulus, you have quite in terms of the firing rate, and then each successive stimulus, it reacts a little bit less to the input. The experiment that was done for stimulus, and we need to go from this firing rate data to connectivity data. So what we did was to bin the firing data by 5 millisecond intervals. Basically, the 5 millisecond interval was chosen so that you wouldn't have any neuron firing more than once within any time bin. And then we need to figure out sort of which nodes are being connected. And so we had what we call transmission response rule, saying what we want to think of in each time bin a certain subgraph, original directed graph of the structural directed graph. And so this time bin, what we would say was that there's an edge, a connection from J to neuron K if there was already a structural connection, so there's a possibility that they're connected. And neuron J spikes in the nth time bin, so this nth period of 5 milliseconds. And neuron K fires in 7.5 milliseconds of neuron J. So the 7.5 milliseconds was chosen after some sort of statistical analysis that I'm not going to go into right now. I think it's better to just go and talk about the results since we're short on time. And since with a time series of different topological variants. So by doing this, this activity, we end up having a time series of, first of all, directed graphs, which then leads to a time series of directed flag complexes. And time in, you can look at the directed flag complex and calculate its number of simplices and so on. So these things are much smaller. They have only maybe sometimes 3 simplices, but usually they're sort of 3-dimensional, basically. And then you can look at the Betty numbers, and you can look at all sorts of things. So in this slide here, we have 1D is the number of edges, 2D is the number of simplices, beta 0, 1, and 2, those are the numbers, and EC is the Euler characteristic. And on the x-axis, we have the time milliseconds, and just recall that we have stimulus incoming every 50 milliseconds. So see this initial spike in the number of 1 simplices, which is basically some sort of proxy for the firing rate. A spike in the number of 2 simplices, although we said that the result reactions are different for the circle and the point. A negative spike for Betty 0, Betty 1, Betty 2. There we see a big difference between the reactions, and the characteristic spikes. We also see a really big difference in the way that the circuit reacts to the 2 different stimuli. Sort of give you some indication that maybe there's something going on with topology. So our point is not to show that topology provides the best classifiers, the best way of distinguishing between stimuli, but just that it's detecting something. It's worth thinking about topology in this context, because it actually is picking up something about what's going on in the circuit. So the Gaussian-based classifiers, in order to see which of these topological metrics was behaving well. So in this diagram, so we're looking at these various metrics we have. So along the x-axis we have time, and on the y-axis it's the percentage of time that you're actually classifying things properly. So we have frame rates, number of one simplices, number of two simplices, Betty numbers, and Euler characteristics. And we'll come back to looking at it in a different way. We use first time period from 0 to 10 milliseconds, doing a pretty good job with the Euler characteristic, the Betty 2, and so on. They're doing a pretty good job of classifying between the two, distinguishing between the two stimuli. Things don't go so well after you get past 10 milliseconds. If you look at the heat maps again of the way it was reacting, you see that, well, okay, if you just use eyes and you look at it, which is kind of a cheat, because computer cannot do that, but if we do, then we see that we can see very clearly between 5 and 10 milliseconds and pretty between 10 and 15 that we have two different stimuli, but starting at 15 to 20, it becomes very hard to see that we actually had two different stimuli. So it's not surprising that the ability of these different metrics to classify things decreases there. And then it goes up again when we have stimulated it again in 15 milliseconds. See the classification. We look at different time bins. So we have 4 different panels here. In the first panel, we're looking at time bin from 0 to 5 milliseconds and see which of these different metrics classify correctly. So we said in the first time bin from 0 to 5 milliseconds, the number of two simplices, Betty 2 all do a very good job of classifying between the stimuli being either the point or the circle. In the time bin, again, got this number of two simplices and Betty 2 and other characteristics, which is doing a fantastic job of distinguishing between the two of them. Between 55, nothing is working too well, though the two simplices and Betty 2 seem to be doing best. And we move 5 milliseconds later, and the number of two simplices and Betty 2 are doing a great job. So the point is not to say this is the ultimate classifier. This is, topology is saying everything about neural activity. It's to say topology at least is seeing something, and we need to know what that something is and why topology has anything to do with it. So to look at this, we can look at it sort of a two-variable classification. In panel A, we're looking at the time bin from 5 to 10 milliseconds, and we see if we compare, do correlations between these different metrics, that basically looking at correlations enables us to distinguish between point and circle. So the dot being the circle stimulus and the triangle being the point stimulus. But if we go a bit further along, in the 200 and 205, that actually in order to distinguish between the two by one of these two variable classifications, we really need to use topology to do so. Okay. So, questions. What is topology of any theory? What are we seeing with this topology? Biological significance. Are these some sort of spatiotemporal coding? So when we're talking about neural coding, one often talks about rate coding or spatial coding, but this seems to sort of take spatial relationships and time into account at the same time. It's a pretty interesting way of thinking about neural coding. Let's compare this activity across the different rat types, because this was just the activity that we did. The functional study was done on the average rat type, and I'm curious about how these differences in structural topology that we noticed from comparing Euler characteristics and Betty numbers, how it will show up in functionally. And there's some experiments that we plan to run soon. Because something about how function is emerging from structure that I think we need to understand. And to introduce, so these reconstructions they've done so far, they call them first draft reconstructions, because there's still a lot more they need to build in. They need to build plasticity in, they need to build gill cells in, they need to build age-dependent change into it. And also, once they have built these changes into the network, which should happen sometime summer, I hope, then we're going to go back and look at the network again and see how this sort of plasticity affects the topology of the network. So thank you very much for your attention, and I'd be very happy to answer any questions you may have. An article about this work is on the archive, and Bill has very nicely shared his code as well for doing simulations. So thank you very much.
Kathryn. Do we have questions for Kathryn?
There were plenty of chat that I just time to read.
So by MV who asked questions. Let's see. Petri, do you think about comparing the corresponding configuration model that is observing directed degree sequence of the original graph?
That's a round comparison that one could do. It's not one that we have done. It's a reasonable one to consider. I suspect that it would still be very different and not as highly structured.
Still not quite clear on all the details of the model. So have these. So there's these 7 hexagons, and each of these is called a microcircuit. Is that right?
So these columns themselves are called microcircuits. So let's see. I think can I go backwards?
So are there 7 columns that are together? Is that?
Yes. So if I go to here. Let's see. And so this here is called the microcircuit. So it's the 6. It's a diagonal thing that's roughly, what did I say, 2.2 millimeters across. There's 6 layers. 1, 2, 3, 4, 5, 6.
Okay, and then you have 7 of those arranged together.
You don't necessarily. Sometimes you consider them all together, and sometimes you consider them individually.
Okay.
But we were studying one at a time.
One layer of 6, or just one of the 6 pieces?
One column of 6 at a time. So a column like this.
Okay.
Considering the continuity among all the layers.
Sections, are they all within one of the layers, or they go through all of them?
No, they cross layers. They definitely go cross layers. Particularly, I mean, usually they're sort of going up. The connections are going from 6, 5, and 4, and so on. I think just go. So here's the microcircuit portal, which is fun to play with. And so here's the 6 layers, 1, 2, 3, 4, 5, 6. And see the connections going between the different layers. We see layer 6 is primarily connected to itself, but it's also connected to 5, connected to 4, and so on. And so there are connections going between the layers like this. And,
so mostly going up, and not exclusively.
Not exclusively.
Okay. Multiple edges between a pair of vertices.
Edges going both ways.
Edges from a vertex itself?
I mean, a loop? Yeah. I don't think so. I don't think that happens logically.
Okay.
So.
All right, very helpful.
Yep.
Thank Kathryn for a really nice talk. Thank you.
Thank you, Peter.